vesicles in immunology. I'm really delighted to welcome Ivana Milic from Aston University, who's going to give us a talk on extracellular vesicles and immunomodulation. And Ivana, I gather you've been a lecturer in biosciences since August 2022 at Aston University. So thank you for agreeing to do a talk, and I shall hand straight over to you.
Thank you. Thank you very much for inviting me. This is a great opportunity for me to share what we have been doing over the past 8 years since I've joined Aston University for my postdoc. So I will be talking today about extracellular vesicles and immunomodulation. And to set the scene, before I go into the science of it, I'm sorry, can you see my, okay, there you go. So I believe that every one of us has at least for once experienced one of these 5 well-known cardinal signs of inflammation. And they're actually the direct consequence of what is going on the cellular level. So if we look at the whole time frame of inflammation from the point of its onset to the tissue regaining its homeostasis, you can appreciate that this whole process is actually driven by the timely infiltration of different types of immune cells to the site of the tissue damage. But these cells need a very specific signals to be able to know when to migrate in and when to back out from the inflamed tissues. And all of this is actually tightly regulated by the bioactive lipids, known as lipid mediators of inflammations, which are synthesized by the coordination of the enzymes and the cell receptors locally at the inflamed sites. And you can see that some of them marked here in red are driving the inflammation, while those marked in green here are the ones that are actually helping tissue regain its homeostasis. And I will talk a little bit more about this during my talk. So the apoptosis here plays one of the central roles necessary for the tissue to actually go towards the root of homeostasis. And I will focus my talk today on the role of extracellular vesicles released during this process and how do they actually help drive or resolve inflammation in that context. So if we talk about apoptosis as a control cell death, the aim of the cells which has committed to apoptosis is actually to effectively communicate their presence to an immune system in order for the tissue damage to be resolved and to prevent the chronic inflammation. And the cells are doing that via the widely known soluble factors, and I'm sure that most of you have heard at least for some of those here on the slide. But what is very interesting that in 2012, for the first time in one of the reviews, the so-called microblasts at the time, which had on its surface ICOM3 protein, have recognized as the functional entities derived from apoptotic cells. So actually, this recognition stems from the research that was done here before my time at Aston University. So if you look at this upper graph here, from the point that the cells commit to apoptosis, they start slowly releasing, exposing the phosphatidylserine on its surface, which is marked here in green, and the annexin 5 staining for these exposed phosphatidylserine is obviously going to increase during the apoptosis onset, but at the same time, cells are losing then their mean cell volume. If we are looking for the ICOM3 specifically, we didn't manage to find the ICOM3 in the soluble factors, which led us to believe that this ICOM3 is effectively getting lost from the dying cells in a form of what we thought at the time was something like a microblast. And when we use the secretomes of the apoptotic cells and place them in a horizontal migration assays against the monocyte-derived macrophages, we have seen that the vesicles from the different type of dying leukocytes were actually able to attract macrophages. When we use the MA4 antibody to block the ICAM3 on the surface of these microblasts, we have seen that this macrophage directionality towards the source of attractant has been lost. Since then, we've learned a lot about so-called apoptotic EVs, and what is important for me to tell you right now is that once the cells commit to apoptosis, they secrete increasingly larger number of vesicles over the course of the apoptosis itself. And this is something that you can see here that is not only specific to immune cells. You can see the fibroblasts acting in exactly the same way. So during the apoptosis, we do not have only the release of so-called apoptotic bodies, we also see increase of the EVs in those small size range, which is usually attributed to the exosomes. And you can see here at the bottom of the slide on the right that actually the mode diameter of these released EVs is not particularly changing. We measured this using QNano years ago, but results are fairly the same when we look at the different models over the past few years. So I would like again to note that there is a tremendous release of small EVs during the course of apoptosis, and there are a few groups around the world that are interested in apoptosis and apoptotic EVs from the different perspectives. And you can see that a certain type of immune modulatory function can be assigned to these EVs. These EVs will interact with the host cells in exactly the same manner, more or less, as any other EV. So they can be taken up via phagocytosis, they can interact with the host cells by ligand receptor interactions and so on, and they will exert this function. So the questions that we are asking is actually which feature of the EV is responsible for the functions that various groups up to date have seen. Are those physicochemical features? Are we talking about the size? Are we talking about the surface charge, perhaps? Are those biomolecular features? Is there a biomolecule that is responsible for all of these effects? Is there going to be a protein? Is there a lipid? Is there metabolite? Is there a genetic material that EVs are carrying? Or is it perhaps due to the fact that the EVs are derived from the different cells or the different cellular subcompartments? And when I joined Aston University, this was actually the first thing that I was tasked to do. So to answer, or at least to aim to answer this question, we had to establish several things. So first of all, we had to be absolutely confident in our isolation techniques. Then we proceeded to the multi-omics characterization of our EV isolates, which helped us to postulate at least what are the key biomolecular features of these EVs, and then attribute certain functionalities of those in a series of functional studies that we have performed in order to define the mechanism of immunomodulation. So one of the very first things that we've done, we looked at the protein profiling of apoptotic EVs. So now here on the left, you can see how we isolated EVs from the peripheral human blood. We used negative isolation. Here you can see the results specifically from the primary human T cells, CD3 positive T cells. We committed these cells to apoptosis, and we have collected EVs 18 hours from the apoptosis onset. If we look roughly at the data, we can see that EVs are specifically enriched in a group of proteins as compared to the apoptotic T cells. So there is definitely preferential packaging of proteins in an EVs as compared to the C cells. But then again, depending on the instrumentation that you have, depending on your technical capabilities, these data sets from the protein profiling can be very, very large. So we're talking here about the data sets that are at least 1,000 proteins and higher. So the question is, how do you narrow down that list from 1,000 different possibilities to something that you can actually analyze in the lab? So one of the things that I would suggest everyone to do is to look down the gene ontology pathways, first characterize your EVs, and then dive into these specific groups that could be enriched and that could actually give you answer to the certain functionality that you're trying to describe. So our apoptotic EVs, you can see that they massively enriched immune responses, cell communication, immune cell migration, so all of these features that were actually of interest to us. So in the sake of timekeeping, I will just propose some of the functional features of these EVs. They are, of course, proteins that we believe are quite important. Those will be the proteins that will facilitate uptake or a communication with an immune system. So don't forget about the adhesion proteins. They would be probably the ones that will support the EV uptake by the whole cells. But there is a number of protein that is known to support immunomodulation, such as MIF or FLAP, or some ABC transporters that are well characterized in the cancer models when we talk about immunomodulation as well. So these would be some of the candidates that could potentially play a role in immunomodulation. I mentioned the molecular aspect of apoptosis from its onset to the homeostasis. So going back again, these bioactive lipids are synthesized locally by enzymatic interaction within the cells. Sometimes it is necessary, especially for those pro-resolving, that we have a subsequent enzymatic interaction of different type of enzymes, so it's not a direct enzyme product. And all of these lipids are synthesized locally by the inflamed tissues, which could express the enzymes as well, or by the immune cells. So they are produced in the sub-femtomolar levels, and as such we needed a very sensitive platform in order to profile their possible presence in the EV preps that we had. And to do something like that, we have set up in our lab a platform, which is a targeted mass spectrometry platform, which at the moment looks at 28 different bioactive lipids, and with a possibility to reach this very low concentrations, which are usually be the ones that they would be synthesized endogenously. And we are more than happy to collaborate with anyone who is interested in doing this type of research. Going back to our results, we have shown that our apoptotic EVs carry pro-inflammatory lipid mediators of inflammation, but they also have a pro-resolving SPM, specialized pro-resolving mediators of inflammation. And when we look at their relative ratios, we can see that actually EVs are massively enriched in the specifically arachidonic acid-derived pro-resolving lipid mediators of inflammation, which can potentially explain why the effects that we have observed were usually pro-resolving. These lipid mediators of inflammation are produced by the enzymatic action. So naturally, we asked ourselves, well, are the enzymes present within EVs as well? And the easiest thing to do at the time was to do a Western blot for the different enzymes. And you can see that this family of lipoxygenases, which is important for the synthesis of the pro-resolving but pro-inflammatory lipids as well, was enriched in our EV preps. The next question would be, okay, well, if the enzymes are there, are they actually active? So we managed to find a commercially available kit that was looking at the total lipoxygenase activity. And you can see here in a green tray that our EV preps showed the lipoxygenase specific fluorescence in this assay, and when the EV preps were mixed with a lipoxygenase PAN inhibitor, we can see that this signal was going down, suggesting that our EV preps do carry active enzymes. And this was, I guess, the crucial point in our research which has happened. And from that point, we started calling these EVs active EVs, which because of this made discovery that we had. Incubation of these active EVs, active in lipoxygenases with the cells that do not endogenously express lipoxygenases for 2 hours only, has proven that based on the activity in the cells, we have proven that EVs are capable of transferring the active enzymes to the host cells. And these enzymes, once transferred to the host cells, were capable of further synthesis of their lipid mediators, as we have shown here based on our mass spectrometry measurements. So EVs are carrying active enzymes; they're actively transferring that enzymatic activity to the host cells that naturally do not express these enzymes, and these enzymes can lead to the synthesis of lipid mediators of inflammation. Lipoxygenase is a family of 3 different enzymes: lipoxygenase 5, 12, and 15. And usually pro-inflammatory lipids their synthesis is triggered by the 5-lipoxygenase here in red. And you will usually read about this 5-lipoxygenase as it is like a pro-inflammatory enzyme, which is not necessarily true. If we looked at the SPMs, those pro-resolving lipids, their synthesis is usually triggered by 15-lipoxygenase, which is usually believed to be a good enzyme. But the presence of 5 or 12-lipoxygenase is necessary for the final synthesis of a lipid. So we modified in-house slightly the commercially available enzymatic activity assay, and we have shown that our EVs carry all 3 lipoxygenases once again, but the highest activity is attributed to the 15-lipoxygenase as the most active enzymes that our EVs carried. When we incubated these EVs with monocyte-derived macrophages in vitro, we have looked at the lipid metabolome of the treated macrophages, and we have seen that actually these so-called M-node macrophages have completely lost the expression of the leukotrienes, which are known to be pro-inflammatory, and in return they have massively upregulated 15-lipoxygenase lipid mediator pathway, suggesting that from this angle, lipid metabolome angle, the incubation of non-stimulated macrophages with an apoptotic EVs, preferentially polarizing them potentially towards the pro-resolving phenotype. We have looked at the same thing, incubating our EVs with the human primary monocyte-derived macrophages. We have isolated these from the blood. We have matured them for 5 to 7 days with the GM-CSF to produce so-called unpolarized macrophages, and then we have specifically induced pro or anti-inflammatory phenotypes of these macrophages by incubating them with either IL-4 and IL-13 to produce M2, so-called pro-resolving macrophages, or with interferon gamma and LPS to produce pro-inflammatory macrophages. And then we have performed flow cytometric surface phenotyping. We have defined the phenotypes for M-node, M2, and M1 macrophages, and then we have incubated them with the EVs for 24 hours. And we have observed here, you can see on this PCA biplot that there are distinct features that we can assign to unpolarized macrophages, pro-inflammatory and anti-inflammatory macrophages, but that our incubation with an apoptotic active EVs has actually flipped the surface the surface phenotype of macrophages very closely to the one that that anti-inflammatory pro-resolving macrophages would have. So that was a good thing, and this is mainly contributed to the high levels of the CD206 and the absence of increase in CD40. If anyone is interested about the specifics of this, we have this assay nicely working in our lab. So again, if anyone wants a little bit more details on this, we are more than happy to share. So a lot of data on the in vitro studies and a lot of functional stuff in our in vitro assays. But the next question was, where we asked whether these EVs can recruit macrophages in vivo. The first thing that we looked on was a so-called model of peritoneal inflammation. So when we talk about the mouse peritoneum, in the mouse which is not challenged, we have a presence of so-called large peritoneal macrophages in the peritoneal cavity, and the tissue is in a state of homeostasis. If you induce inflammatory response, a series of events will happen within the peritoneum, with first one being the disappearance of the large peritoneal macrophages. But this whole cycle will finish with the peritoneum reinstating homeostasis, which you will see by the LPM, by the large macrophages appearing again within the peritoneum. When we have injected our EVs into the peritoneal cavity of the control mouse, we have observed that actually almost 2 hours post-injection, we have a large migration of the large macrophages to the peritoneal cavity, suggesting that EVs are more than capable of recruiting macrophages in vivo. One more example, if we look at a slightly different model, so we are talking here about the tumors. So tumors are known to secrete EVs, and these EVs are communicating with the immune system. Here we have an example of macrophage. So if we using a xenograft, if we look in the mouse where we have delivered where we have xenographed the tumor, which is in this case ICAM3 positive, so this was a Burkitt's lymphoma. The hypothesis is that the tumor will grow if there is a recruitment of macrophages due to EVs, which are being released from the tumor and carry an ICAM3. So this is the protein that we already know that plays a role in recruitment of macrophages. If we block this ICAM3 using an anti-ICAM3 antibody MA4, we can hypothesize that the tumor will shrink, because ICAM3 will be blocked and it will prevent ICAM3 positive EVs from communicating with the macrophages, which will lead to the tumor shrinkage. And actually, when we look at the number of recruited macrophages within the tumor, we can see that this is exactly what has happened. So ICAM3 on the EVs was blocked. The tumor has stopped growing as aggressively. And when we look at the tumor size, we can actually see that there is a significant reduce in the tumor size compared to the mouse that have been injected with an isotype control. And at the very end, I would like to tell you that we are also taking a different direction, knowing that EVs carry enzymes, that they carry enzymes in their active form, and that these enzymes can actually potentially play a big role in immunomodulation. We have looked at a different class of enzymes from the sphingosine rheostat, which is also quite important in synthesizing the anti-inflammatory and pro-resolving lipids of inflammation. And we are trying to model this for our further research in the glioblastoma. These specific lipids and the enzymatic machinery is highly enriched in brain, and could potentially play a role which could explain why these tumors in particular are so fast growing and very hardly curable. And with this, I would like to thank everyone who was involved in all this research over the past years. There are some lab members that are no longer with us. We have some active collaborations with different universities. We are thankful for the support that we are getting usually from PBSRC, Alzheimer's Research, which has funded a lot of the research that you have seen here. I would like also to say that we are recruiting in the next couple of months for an 18-month-long project. So there will be a vacancy for a postdoctoral position as well as for the technical support. So if you're interested, keep an eye on Aston webpage for the job vacancies, or contact us directly and we can chat about it. And thanks again.
Brilliant. Thank you so much, Ivana. That was really thought-provoking and very interesting. So thank you very much. I think as we have slightly run over on time, I think in keeping ourselves to time, I think if it's okay, if people put questions in the chat, and if you're happy to answer them via the chat, if that's okay. Yeah, so people will put questions in the chat for you if they've got any questions. So thank you very much.
Thank you once again.
No, thank you very much. Okay, so we'll move on to our next speaker, which is an early career first of our early career researcher speakers in the EVs and immunology section. So it gives me great pleasure to introduce Nancy Gomez, who is an international PhD student who actually works in my group, and she's going to talk to you about the effects of cigarette smoke and e-cigarette vape on the production of extracellular vesicles by immune cells. So Nancy, I'll hand over straight to you. You're on mute.
Yeah, thank you for that, Lucy. Just trying to share my PowerPoint. Sorry about that. So yeah, can everyone see that?
Yeah, we can see that. Thank you.
Okay, yeah. So as Lucy said, my research involved investigating the effects of e-cigarette vapor on the production of extracellular vesicles by cells of the immune system. Now, we all have an idea of what EVs are. They are these important intercellular communication mediators that are able to transfer biological signals, including lipids, nucleic acids, and proteins enclosed in a phospholipid bilayer, which allows them to travel far distances from their cells of origin. Now, early in my PhD, I did a systematic review looking at extracellular vesicles and chronic obstructive pulmonary disease, or COPD. And COPD is defined as is characterized by irreversible airflow limitation. And when I did the systematic review, one of the things that we noticed was that not a lot of studies looked at EVs derived from cells of the immune system and mainly focused on endothelial or epithelial cells or circulating EVs, which didn't classify where they came from. And one of the things that we know about COPD is that cigarette smoking is a risk factor. So my PhD started to look at what is the effect of cigarette smoke on the immune system and how they're producing these EVs. Additionally, we know that cigarette smoking has been looked at as a safer alternative to cigarette smoking. Sorry, I said e-cigarettes. E-cigarettes are looked at as a safer alternative to cigarette smoking. However, research hasn't fully elucidated the effects of e-cigarettes, both acute and long-term effects. So we wanted to compare both cigarette smoking and e-cigarette vapor on the effect of the EV production by cells of the immune system. So next. So we set up this model in the lab where we're able to isolate. Get our EVs from PBMCs that we isolated from fresh blood, which we obtained from volunteers. So we had a PBMC culture, and that was exposed to cigarette smoke. And we used different techniques to observe our EVs, including imaging flow cytometry, TM, NTA, and spectral flow cytometry. As previously mentioned by my colleague, Davis, we also use calcine AM, where it works by being internalized by the EVs, and it's cleaved by esterase, where it becomes fluorescent and is EV impermeant. So we developed this model and then applied it to both cigarette smoke and e-cigarette vapor. So when looking at our EVs to confirm their structure and morphology, we isolated our EVs after culture by size exclusion chromatography. And we used the original 35 columns, which allows us to obtain EVs below 350 nanometers. we first observed by transmission electron microscopy, and here we can see typical cup-shaped morphology consistent with the morphology of the EVs. We also did nanoparticle tracking analysis, where we confirmed that most of our EVs were under 200 nanometers, with the size, with the mode being 128 nanometers. So next, we then focused on looking at the imaging flow cytometry. Again, our PBMCs were cultured up to 48 hours, and we had them under different conditions. So we had our control, which was no exposure, our 1% and 3% cigarette smoke extract, or CSE, 1% and 3% e-cigarette vape extract, or ECVE, and our 2 microgram and 200 micrograms per mil of nicotine. Which, we know that we find nicotine in both cigarettes and e-cigarettes. So after we cultured them, we also added calcine AM and our anti-tetraspanins antibodies, which were specific for CD9, CD63, and CD81. Right, so previously mentioned as well, one of the first things we did is use size beads to kind of create a gate that we wanted to focus on our EVs. And here we wanted to focus on small EVs, particularly 200 nanometers and below. So created a small gate, a gate for small EVs. The optical resolution cutoff of the image stream is 200 nanometers. So anything below that wouldn't give you a bright field spot. So, and yeah, so we wouldn't expect, so we gated for 0 bright field to not get an image there. We also gated for 1 calcine spot count we could obtain, so we could remove any coincidental events. And here we can see the green color of the calcine, and we could collect our statistics. Now, our results showed that that there were no significant differences in the number of EVs being produced by our PBMCs under any of the conditions, both compared to the control and to nicotine. So the next thing we looked at was the tetraspanin profile of our EVs. And along with my supervisor, David, we created a series of gates that helped us to look at our tetraspanin expression, particularly looking at if EVs express 1 tetraspanin, our double positive populations for expressing 2 tetraspanins, on our triple positive populations, which we could see if there were some EV subpopulations that were expressing all 3 tetraspanins. So here we can see that you have the calcine, CD63, CD9, and CD81. So when looking at, under the different conditions, we did find some significant differences in the tetraspanin expression of the EVs, particularly for CD9, and that would have been for 3% e-cigarette vape extract compared to the control, and as well compared to the 200 microgram per mil of nicotine. This was regardless if they're expressing 1, 2, or 3 tetraspanin, just the CD9 expression. But we also looked at subpopulation of these EVs, and what we did find that for CD9 positive only, so EVs that were expressing only 1 tetraspanin and that being CD9, there were some differences between the EVs that were expressing that for the different conditions. So that in particular would have been an increased expression for 3% ECVE compared to the 0% control and compared to 200 nicotine. There were also some, we could also see that for CD9, we had some increase after 1% and 3% e-cigarette vape extract compared to the control. And it was also particularly interesting that the 3% for e-cigarette vape extract was also increased for its matched cigarette smoke extract concentration, which is 3%, and for the 200 microgram of nicotine. And then we also had a significant difference of that 200 nicotine compared to the 3% e-cigarette vape extract for CD63. So some differences between the tetraspanin expression. If we look at CD81, there wasn't really any changes, which we have seen previously as well, so it was expected for that tetraspanin. So aside from looking at the tetraspanin, We also wanted to look at some at the expression of some membrane markers found on the EVs. So we use a MaxPlex exosome kit, and the way the kit works is that it has 37 different markers. It has 39 beads, but 37 markers and 2 control. And using flow cytometry, here we can see we gated for the 39 populations. This is our buffer, and this is one of our samples. So what we observed were some small, non-significant differences. And how we obtained this result is by getting the median APC signal. And what we did is we subtracted the median APC signal for our 0% control from each of these, from the matched samples to get this heat map. So we saw some small, non-significant changes, and we can see that there was some down regulation of some markers and some upregulation of other markers. But again, here we only had an N of 3, and there could have been some variability between our volunteers that we had that gave us blood for this part. So aside from looking at EVs, we also wanted to look at the PBMCs that were producing these EVs. So we did some spectral flow analysis. Again, under the same conditions, this was occurring at the same time when we were getting the EV numbers, and we used a 14-color panel to look at the markers being expressed on our cells. Additionally, we had some cell populations of interest that we wanted to look at, but we also wanted to look at some previous markers such as CD29, HLA-DR 40 and 49E that we had previously identified using the MaxPlex kit that were of interest, and we wanted to see using this panel if there were also changes in expression in the cells. So I did some manual gating, but just in the interest of time, I'm only going to show the clustering analysis, which is a more objective or non-biased form of doing the analysis. And here we can see with the Tisney plot, we were able to identify 15 meta-clusters doing this clustering analysis. And in particularly population 7 was a very high population in the PBMCs, followed by population 0. Here we can see the different expressions of the markers in the clusters, and in particular looking at the higher populations, they would have been our CD4 and our CD8 T cells. So they were clustered together. We could also see the expression of the different markers on our PBMCs. However, what we found when we did the statistics was that there were no significant differences in these meta-clusters for any of the conditions that we had exposed them to. So in conclusion, just to summarize, is that the number of EVs being produced by our PBMCs did not significantly change under any of the conditions. However, we did have some significant differences in the tetraspanning profile of EVs, particularly for CD9 and CD63 between the conditions. We also saw that the protein and the surface marker profile of EVs were affected by the CSE and ECVE treatment, and this was compared to the control. And we saw that the clustering analysis identified 15 clusters present, but showed no significant differences between the clusters. What we think is that the CSE and started cigarette smoke extract and the e-cigarette vape extract may possibly modify the EV profile in humans, and possibly the exposure to these components can result in changes within the cells that are producing the EVs and consequently affect EV formation. So yeah, so that's me. Thank you.
Thank you very much, Nancy. You've run right to time, so in the interest of time, if anyone's got any questions, please put them in the chat for Nancy and we'll move on to the next presenter. So next up is Amon Hackney, who is from Montfort University. He's currently a final year PhD student, and his talk is looking at fish oil supplemented treatment. and how it affects the nature of extracellular vesicles in pancreatic cancer. So I'll hand over straight away. You're on mute. Sorry, we can't hear.
There we go.
Ah, that's it. Well done. Thank you.
Is the presentation up for everyone?
Yeah, we can see it.
Yeah, perfect. Okay. As Lucy said, my name's Amon Hackney. I'm a final year PhD student at Montfort University. And I'm going to be talking to you today about EVs and pancreatic cancer. So pancreatic cancer is a really horrible disease. It's the 5th highest cause of cancer mortality in the UK. It has the lowest survival rate of any cancer and a very poor prognosis, usually less than 6 months from diagnosis. It's highly aggressive, tumorigenic, and there is a lack of early warning signs. People usually get really generalized symptoms such as stomachache, and it's really hard for a doctor to diagnose early. Usually once it's diagnosed, it's already advanced and not suitable for surgical resection. And the chemotherapy for pancreatic cancer is really poor. Hence the high morbidity, and it's also associated with venous thromboembolism, which increases the morbidity even more. It's previously been shown that omega-3 fatty acid supplementation of the standard gemcitabine therapy is associated with anti-inflammatory status and increased progression-free survival in these patients. It's also important to note that intracellular communication is crucial for metastatic progression in this disease. So extracellular vesicles in pancreatic cancer, as we all know at this point in the day, they're biologically active nanovesicles released by cells. They can contribute towards disease pathogenicity, progression, and they're also involved in thrombosis. In pancreatic cancer specifically, they've been shown to facilitate growth of the tumour itself and also metastasis at distant sites. It's been shown that the circulating count of EVs is increased at later disease stage. And as I mentioned earlier, PC is associated with venous thromboembolism, and patients that are diagnosed with VTE also have a shorter overall survival and progression-free survival. The hypothesis of our study was that omega-3 supplementation of chemotherapy will reduce the circulating number of extracellular vesicles and also reduce their prothrombotic phenotype in pancreatic cancer patients. So what did we do? We had a cohort of stage 3 and stage 4 pancreatic cancer patients, all receiving gemcitabine chemotherapy. This was once a week for 3 weeks, followed by a week off on a cycle. The two groups were split, so some had just the gemcitabine, and the trial group had gemcitabine immediately followed by a 10-gram infusion of omega-3 fatty acids. We isolated the EVs from these patients using serial centrifugation, and they were split into 5 groups, sorry, the healthy control, stage 3 patients receiving gemcitabine, stage 3 patients receiving the supplementation, and then the same for stage 4. The circulating number of these EVs and the prothrombotic phenotype was assessed using flow cytometry and staining. So first things first, the total EV counts. We found that the EVs were significantly increased at stage 3 following with the supplementation. However, at stage 4, this was not significantly different from the healthy control. Whilst conducting this work, we found that there were 2 distinct populations of EVs. We called these high forward scatter and low forward scatter. And the literature showed that the smaller low forward scatter EVs are associated with disease progression and cellular dysfunction. Again, we found that the smaller EVs were significantly increased in the supplemented group at stage 3, but then at stage 4, again, this was not significantly different from the control. We then looked at the prothrombotic EVs, so we stained them for tissue factor, which is a procoagulant initiator of thrombin formation. It's been shown to promote tumour growth, angiogenesis and metastasis, and in pancreatic cancer specifically, high levels of circulating soluble tissue factor have been associated with adverse prognosis and increased tumour grade. Again, here we found that at stage 3, the supplemented group had higher tissue factor staining than the healthy control. And again, at stage 4, the supplemented group was not significantly different from the healthy control. Next, to try and make sense of those numbers, we looked at tissue factor activity, which is the actual ability of that tissue factor to convert factor 10 to factor 10A, which is involved in clotting, and it's increased in cancer patients with venous thromboembolism. Unfortunately, we did see a significant increase at stage 4 in the supplemented group compared to those receiving traditional chemotherapy. Obviously, this is not good. If anyone has any hypotheses as to why this happens, that would be great, because we really don't know why. Finally, we looked at adhesion molecules on these EVs. So we found that CD11B positive cells promote angiogenesis and metastasis in pancreatic cancer. And here we found that the stage 4 traditional chemotherapy patients had higher CD11B staining than the healthy control. But again, the supplemented patients were not significantly different from the healthy control. So in conclusion, omega-3 fatty acid supplementation in pancreatic cancer reduces the overall EV count at later stage, reduces the number of smaller EVs at later stage. It reduces the number of tissue factor transporting extracellular vesicles in the later stage, and it also reduces the number of adhesion molecule expressing EVs at later stage. However, as I mentioned, the tissue factor activity does increase in the later stage PC patients with omega-3 supplementation. Obviously, further work is required to assess the effects of omega-3 supplementation and the role of EVs in the inflammatory profile, disease stage and progression-free survival of these patients. Thank you very much. That's all from me. Is there any questions?
Thank you very much. So I've got some questions. Matt, is there anything on the chat?
Yeah, there's a comment, probably more than a question. Did you want to go first, or do you want me to?
Okay, I can go first if that's okay. So really good talk again, another good talk. Thank you very much. I might have missed it at the start, and I do apologise, but what samples were you using to identify your EVs in?
It's patient plasma that was taken immediately after each treatment that they had.
And so, and again, sorry if I missed this, and how did you actually define them as being extracellular vesicles?
So we used a previously defined serial centrifugation method to separate these out. And then we gated them using their forward scatter, side scatter, and compared to some megamix beads of the correct sizes.
Okay, and just out of interest, what flow cytometer did you use?
It's a Curie C6.
Right. Okay. Thank you. Thank you. Sorry, I'll let other people have a chance now, Matt.
That's fine. And then there was a comment. Amy and I definitely jump in and have a little look. So, comment from Alison around fatty acids may be affecting the lipid composition of the membrane. So that could be part of it. And she said she's happy to advise on how to test this. So, yeah, yay the network straight in. People helping people. So yeah, feel free to connect, or we can share details after if you want.
Thank you.
Brilliant.
Just gonna double check the chat, but yeah, nothing in the chat, so all good. Thanks very much.
Great, thank you very much, Amon. That's brilliant. So our last speaker of the day, Rachel Butler. Thank you very much for holding on everybody still for the whole session. So Rachel is a final year PhD student in Professor Andrew Devitt's lab at Aston University, and she's going to be talking about defining the surface proteome of apoptotic cell-derived extracellular vesicles. This is a nice follow-on and a nice talk to finish the session. So thank you very much, Rachel.
Great, thank you. So I'll just share my screen. So let me know if you can't see that. So yeah, thank you for the introduction. So for the sake of time, I'm just going to dive straight in. So our group as a whole are interested in how both viable and apoptotic cells communicate with and modulate the immune system. And in particular, we're focused on the innate immune system, so that first line of defense. And obviously they can do that in a number of ways, but one of the ways that they do that is via the release of extracellular vesicles. We're particularly focused on how extracellular vesicles interact with and modulate one particular type of innate immune cell, the macrophage, which is known for its highly efficient eating capabilities, which becomes particularly relevant in processes such as apoptotic cell clearance. But also they're an interesting cell type because they can exert these two polarized phenotypes. So they can exert these pro-inflammatory functions or these anti-inflammatory pro-repair functions. So my project is specifically looking at vesicles derived from apoptotic cells, and I'm particularly interested in the surface composition of the vesicles. As this will be the first point of contact between a vesicle and a recipient cell. So I'm really interested in those early interactions between vesicles and then macrophages, as Ivana nicely introduced, that they upregulate these pro-repair macrophages, but we don't know how they do this. So if we can upregulate those pro-repair macrophages, then we might be able to think of therapeutic interventions for chronic inflammatory diseases. So that's sort of the idea behind my project. So the first part of the project was to characterise the vesicles that I was collecting in the lab in line with the MySEF guidelines. And one of the best ways to do that is to look at them. And so we did cryo-electron microscopy, and this was in collaboration with Saskia Bakker, who spoke earlier. And we can see that our vesicles have a diverse size range and also shapes, depending on the cell source that they come from. They can appear densely packed with protein, whereas others look empty. And some of them have these multi-lamellar features as well. By nanoflow cytometry, I can see that the vesicles carry these common EV markers, the tetraspanins, at the surface. And they sit between 80 and 100 nanometers in size, with EVs taken from a later apoptotic time point, showing this shift in size distribution as the cell begins to lose its membrane integrity and starts releasing bigger vesicles. So the next part of the project was to try and unpick the molecular composition of the vesicles. And to do this, the group previously did mass spectrometry of vesicles taken from 3 different immune cell lines, the Mu2 B cell line, Jurkat T cell line, and THP1 monocyte cell line. So these cells were induced to apoptosis, and then EVs were collected at different apoptotic time points. And we can see that 414 of the proteins were conserved across the 3 cell lines. We also see that proteins were differentially expressed at each time point. And when we do gene ontology analysis, we can see that some of these biological processes that are implicated in things like apoptotic cell clearance, a lot of these proteins associated with these processes are enriched at that early apoptotic time point. So that's kind of interesting, suggesting potentially an apoptotic cell has some sort of time constraint where it has to really quickly signal to its surrounding immune cells. So I went through a selection process of all of these thousands and thousands of proteins in the mass spectrometry data to pick out a panel of proteins that were predicted to be surface associated and also implicated in these biological process terms. And so I came down to these 4 proteins, CD31, CD47, CD49D, and CD166, which are all implicated in these types of processes like adhesion, migration, and a couple of them also are known for having these inhibitory don't eat me signals as well. So the first thing I wanted to do was look at the expression of each of these proteins in cells. So looking at how the expression of these proteins on the surface changes in cells during apoptosis. And to do that, I did flow cytometry, where I dual stained my cells with an apoptosis marker in Exin-5 and an antibody targeting one of the proteins of interest. So here I'm showing CD31. And so as the cells progress through apoptosis, as shown in the green, we see a significant decrease in the surface expression of CD31. And I see the same sort of trend with all 4 of the proteins. So the question we were asking then was where or how is the cell losing the expression of these proteins, and where do the antigens go? And so, of course, one of the explanations for that would be packaging and shedding via vesicles, which we already know because these proteins were picked from the mass spectrometry dataset, which was done on vesicles. So then the next question to ask was, where are they localized in or on the vesicle? So are they inside the vesicle in the lumen, or are they exposed at the surface and therefore accessible to immune cells? To do this, I did nanoflow cytometry. So here I've just incubated my EVs with a primary antibody targeting the protein of interest. And I did this using the NanoFCM platform, which we have in our lab as of 2 months ago. And I can successfully detect each protein at the surface of the EVs, depending on the cell derivative. And essentially, this is telling me that, yes, the proteins are there, they're accessible, and then could potentially exert a function with a recipient immune cell. So the final part of the project is very much ongoing and is trying to actually characterize the function of each of those proteins by blocking their activity. So the first sort of assay I'm going to show you is the vertical transwell migration assay, which is a very simple sort of setup where you seed macrophages in the top of the transwell and count them as they migrate through the transwell towards this chemoattractive EV-containing secretome. And then doing this, repeating this in the presence of blocking antibodies for each protein. So for the first protein of interest, CD166, I don't see any difference in the migratory profile. You can see that most of the cells have migrated between 4 and 5 hours, and that in the presence of a blocking antibody against CD166, I don't see any significant difference. But for the other proteins, I do start to see differences in the migratory profiles, suggesting that they are potentially involved in that attraction process somehow, but that needs further investigation. Another type of assay that we've established is the ability to measure cells taking up EVs. So I've stained here vesicles with the Bo-Dippy maleimide dye, which binds thiol groups on proteins, and then incubated those vesicles with macrophages and measured the cells over time, seeing that as they take up the vesicles, they themselves become fluorescent. And so I'll repeat this as well in the presence of the blocking antibodies to see if that has any effect. And then finally, this is just recapping the work that Ivana showed previously, where we have a well-established model for macrophage phenotype for primary cells. So I'm going to try and replicate this in cell lines to gain a more high throughput version of this model to then go in and test whether in the presence of blocking antibodies, macrophages still skew towards this M2 pro-repair phenotype, or if they don't, and it actually changes that. That is my talk. Happy to answer any questions now or in the chat. And yeah, thank you to my supervisors, Andrew Devitt and Alan Goddard, and also my NanoFCM colleagues for all their support, and to Ivana for the donation of the mass spec data sets.
Really good. Thank you very much. A really great last talk. I've got a couple of questions. I'll just ask Matt whether there's anything on the chat.
Not yet.
Okay, so I'll go first. So a really interesting data set. Thank you very much. I thought apoptotic EVs, and again, you have to correct me here, I thought apoptotic EVs were larger than the sort of smaller extracellular vesicle sizing that you mentioned. I thought they were much greater in size.
No, so you can, of course, look at the large apoptotic body population, but here I've spun those out through my isolation process, and I'm just focusing in on the small EVs that apoptotic cells release in a great number, particularly when they're undergoing apoptosis versus viable cells, as Ivana showed before as well.
Okay, thanks for clarifying. And do you think it's going to be a combination? So obviously you identified some potential markers on the surface of the EVs for receptor-mediated intake, but do you think it's going to be a bit of a balance between just simple sort of phagocytosis, just testing the microenvironment, and specific receptor-mediated uptake?
Yeah, I think it's not going to be as simple as one golden protein of interest, unfortunately. I think it's going to be a combination of things. And as Ivana showed, that there's all the lipid mediator side as well. And I'm not even looking at the cargo that these EVs contain and how that would modulate macrophage phenotype either. I'm just trying to focus in on those early surface interactions and whether I can detect any mechanism behind that.
Do you know, I think the more we all get into EVs, the more we realise it is complicated and there is no simple answer. And there's so many different things that influence. And I think your talk is a really good exemplar of you know, the exciting things that you can find from EV research, but also the challenges that come with doing EV research. So thank you very much, Rachel. Really great talk. And I'd just like to, before I hand over to Matt, just say thank you to all of the speakers in that session. I think everybody did a fantastic job, and we've still got lots of people in the meeting. So thank you everyone for sticking with us. And I'll hand over to Matt now for discussions about what next with this network.
Yeah, thanks, Lucy. And yeah, I guess just to reiterate your comments, I think EVs are technical enough, and then EVs within immunology is like complexity on complexity. So yeah, so ultimately that draws a close the speakers that have kindly brought their research and their interests forward. And really, just for those that are on the call, there's an opportunity to provide feedback and plans moving forward, really. So this was started out of enthusiasm and energy on our part, and I think I potentially speak for Lucy as well when we're both blown away by the sort of support and interest that there's been. Hopefully you've all enjoyed it. We're very keen to have some feedback and for everyone to input on what this network looks like and how it goes forward. So we've discussed the idea of an in-person meeting. If keeping it virtual on an afternoon makes more sense, please feed that back. And yeah, there are opportunities for feedback directly to Lucy and I, or through Alex, or even in the chat, or if you wanted to jump on and have a conversation now, we'd be open to that. But other than that, just to say thank you to everyone, really. And this isn't hours that we're imposing on people. It's very much if you want to hear from Big Pharma about their interest in EVs, we can look to contact them. If you want to hear more about MSEs as a model, then we can kind of look at that as themes, and we can kind of theme it and do it that approach, or we can yeah, like I say, open to the network for contributions. And this is a groupthink approach, I guess, not to
Yeah, I might just add to that, Matt. I think, you know, I'm involved in multiple networks within the Midlands region, and so I think really probably a call out from both Matt and myself is to say any young enthusiastic people on this call that are interested in doing EV research and want to get involved in being part of the Midlands Innovation EV Network, again, you know, do get in touch with us. We really want this to be driven by the researchers undertaking EV research. Like I say, we've done lots of meetings, we've done lots of conference presentations, and really the reason that the focus today was on the early career researchers, although we had obviously two fantastic keynote talks in both of our sessions, but it was really important that we get early career researchers talking about EVs and the research that they're doing, so we can build that collaboration across the Midlands region. So please do, you know, not just to give us suggestions about future events, but also get in touch if you want to be involved in being part of this Midlands EV network. We would very much welcome you on board, because at the moment really it's pretty much Matt and I. So yeah, Alex, you've got your hand up.
I was just going to say there's also different things that we could look at, peer support networks, whether we could do some shadowing, there's training opportunities. It's really think outside the box with where we could go with this. We're trying out different models with this network. It's not your everyday MI network. So think what you need and let us know.
Yeah, and I see, Alison, thank you. Face-to-face meeting would be good for networking. I agree. I mean, I think we've kept all of the Midlands Innovation Immunology meetings as online meetings, because obviously we also have the Midlands British Society for Immunology group, and we do an in-person immunology meeting for that. So we thought, because this is being launched via the Midlands Innovation Immunology Network, we'd have it as an online meeting. But really, for the EV network, it can go anyway. So if we feel like a face-to-face meeting would be more beneficial, which I think I agree with, then certainly something we can think about for next year's meeting. I think if there's no more comments to add, I think it really lands on me just to say thank you so much to all of the amazing speakers today. I think it's been an absolutely fantastic afternoon, learning about all of the EV research that's going on within the Midlands. So thank you to every single one of you that's done a presentation. Thank you to all of you for staying for the entirety of the afternoon. We really appreciate that. Sometimes when you're the last talk of the day, it can be quite depressing when you see there's only 10 people left. So the fact that the majority is still here is absolutely fantastic. And I think at our peak at the very beginning, I think there was about 86 attendees, which is just fantastic for an online conference talking about extracellular vesicles. So thank you very much to everybody for joining us. Please do get in touch, send us any suggestions, let us know if you want to be involved, and hopefully we might see you in person next year at the next Midlands EV Network meeting.